So it's an October-November paper, variant 2. Okay, yeast is a microorganism that can use the sugar sucrose to obtain energy for growth. When yeast is added to sucrose solution, it grows, producing bubbles of gas. Okay, this would be anaerobic respiration then. Okay, and anaerobic. Which gas is produced in anaerobic respiration by yeast? Hmm, correct. Okay. There are four test tubes. Okay, he started with a supply of 5% sucrose solution. He labeled four test tubes. What would you use to label the test tubes? Marker. Okay, good. He followed the procedure at 15 cubic centimeter of 5% solution to test tube A, then 9 cubic centimeter of sucrose solution and diluted with 6 cubic centimeter of distilled water to make a total of 15 cubic centimeter. Now it would be 3%. Okay, then we have added 3% of sucrose solution and 12 cubic centimeter of water. So this solution would be of 1%. Clear? Okay, we have diluted it, we have changed the concentrations. He measures the volume of 5% sucrose solution and distilled water with a small measuring cylinder. Describe how he could ensure that the dilutions produced were correct. Okay, wait. Since he using a single measuring cylinder, so but he has only one. So he would clean it every time. He would rinse it or he would clean it every time to ensure that there wouldn't be any transfer of material, like there shouldn't be any solution inside it when he made a new dilution. Okay. S- no, 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 wait, you're not getting me. Okay, I'm using a single glass, let's say. So when I'm transferring the content, there may happen that 2 to 3 drops left over there. So when I add a new solution in that, my readings can be disturbed, right? So I need to clean the test tube first so that the new content should be added. But still, distilled water is there. Distilled water can destroy it. You have to like dry it up. Okay, clean and dry cylinders. Okay, okay. Water bath is used and the temperature is 40 degrees Celsius. How do we maintain the temperature? What thermostatically controlled? Okay. He added 1 cubic centimeter, oh sorry, 1 gram of yeast to each of the test tube. One is 5%, other is 3%, and the next one is 1%. Okay. And the fourth, oh wait, I have missed the fourth one. The fourth one has distilled water. This would be used as control. Okay, the last one has a 0% solution basically. Okay. The four test tubes were placed in hot water. He decided that any change in the height of mixture in each test tube would be an indication of the change of its volume. So how much the yeast has grown. So these are the few readings he has given at 5 minutes and at 10 minutes. Okay, what is the percentage of A? 5%. Next is 3. Don't add the sign of percentage as it is mentioned over here. Then 1 and 0. Wait. These are the one. Na? Test tube A is 5%. Test tube B is 3%. Test tube C is 1%. Okay. Okay. So percentage has given. Okay. What should be heading over here? What has he observed? It's written over here. Total increase in height. And what would be unit? Read the statement again. Okay. Okay. First is 40, 26, 20, and 5. Next is 90, 64, 44, and 5. Okay. Use his result to describe the effect of sucrose concentration of yeast growth. When there is more concentration, you can see there is more change in height, right? So you have to conclude this result for 3 mark. Do it. Uh no, you have to explain the result. Yes. They are not asking why we are using control. They are asking uh, was same. It was 5 in both. So why it's showing, why it's giving 5 value? Wait, it contains yeast, but a very little sucrose. Wait, yeast is added same in all test tubes. Very less. Wait. Yeast also has the starch. Yeast itself has a small amount of starch. Hmm. It is. Wait. Yeast is a cell. A cell can have a sucrose of its own. Simple. Even though solution has 0% sucrose, but still some sucrose is present in the yeast cell. That's why the result is 5. It isn't 0, it's 5. Yes. Okay. Okay, he wanted to carry out the investigation at a temperature 40. The thermometers in the diagram show the temperature. What is the temperature at E and F? Shall I make it bigger? Sink again. No. 38.5. And the second one is? Yes, I can. 27. So it's 38.5 and 27. And what are the units? You haven't mentioned the units. Degree Celsius. Okay. Okay. Suggest how the temperature of the yeast mixture could have been maintained at 40 degrees Celsius throughout the experiment. What have you used? Okay, thermostatically controlled water bath. Okay. And how do you know the temperature is 40 degrees Celsius? We use a thermometer to check whether the temperature remain throughout same, or it may happen that the heat escape out and the temperature falls, so you need to check it at regular intervals to maintain a temperature. Okay. Okay. Yeast uses sucrose to obtain energy for growth. Suggest a simple method to demonstrate that this process requires enzymes to be present in the yeast. So you need to have a control to test this, that if enzymes are needed or not. You should take a control. And what would be control? Where enzymes are not present or where enzymes are denatured. Okay, so we you can say that we can boil yeast to denature the enzymes, and it can be observed that there would be no growth, since there is no aerobic respiration. Sorry, there won't be any respiration. Just write respiration. So there won't be any increase in the height. Okay, 3 points. Number one, the yeast is boiled and would be taken as a control. It would denature the enzymes, due to which there won't be any respiration, so no growth or no change in the height. Okay, how do you draw a graph? Exactly. For what? So how many, okay, read this and tell me how many lines would be there. Yeah, you have to make a one light. And wait, you have to make a line graph. They have written it very clearly. Line graph. Hmm. You have to give a key. Like this line is for light, and let's say this line is for dark. Okay. Exactly. And you have to give the key. Keep this thing in mind. Like dotted line is the light one and the solid line is the dark one. No, no, please don't use that. Okay. Okay. Use the data. Okay, use the data to suggest what the student could conclude about the effect of light and dark on the germination of seeds. Here, use this data to explain the effect of germination in light and dark. Hmm. Why? Where do you sow the seed? In the soil. It's dark over there. No. We have studied that the radicle grows first, right? The root grows first, so it doesn't need light. Root doesn't need light. Okay, just explain this one. So we say that germination started earlier in the dark as compared to light, and what can be written after 24 hours? Hmm. Why don't you give value for 24 hour, where one is 0 and another is 5? Okay, okay. So check the marks of this question. This is of only 2 mark. Okay. Hmm. Okay. What two variable other than light are required to be controlled in her investigation, or what should be constant? Okay. pH. What else can be? Temperature. What else can be? Instead of pH, water is required for germination. What is required for germination? Water and temperature are the first thing that you should write. Water. First of all, first of all, it increases in size. The testa increases in size and breakdown. Okay, because of water. So just one reason for using 20 seeds. Correct. Okay, a diagram is given. You have to make this diagram and send me as well one more time. Okay, and then calculate the length and then send me. Okay, describe how would you find out whether a sample of pond water contained this plant. What you would use? You have pond water. Now, how you would prove that this can be present in the water? Correct. Hmm. Okay. Wait. Sample of pond water would be taken. Wait. No use of I and U. Okay. And observed under microscope by preparing a slide. Okay. So this is how you use it. That's why we are practicing it. Okay. Okay. We are done with two papers. It's enough for today.